Hello and welcome to the QIAGEN Antimicrobial Resistance Summit. My name is Andrew Olson and I'll be your host and moderator for today. Antimicrobial resistance, or AMR, detection and surveillance has become a high priority in both healthcare and environmental settings for the safety of patients and the general public. However, samples used for this type of monitoring, such as stool or wastewater, can be challenging, from which sample prep to use, to the sequencing, or to the bioinformatics. We hope to clear that up for you in today's discussion. We are recording today's session, and all participants will receive an automated message with a link to the presentation, but I encourage you to stay until the end for our Q&A session. Along the way, if you have questions, just put them into the Q&A box, and we will do our best to answer as many questions as possible at the end. Also, I will periodically post a quick poll to gauge the interest of each section for future program development and other uses. So let's try a quick poll right now just to see how people are feeling today. So personally, I'm feeling great today. It's a beautiful April day and looking forward to today's exciting topic. So just a quick legal disclaimer here. So the QIAGEN products shown here today are intended for molecular biology applications. They are not intended for the diagnosis, prevention, or treatment of diseases. Please refer to the list of materials for any additional details. So today we will take you on a journey from sample to insight. As you can see, our first speaker is Dominic O'Neill. Dominic has over 20 years of experience in the biotechnology industry. Dominic joined Diagene, which later became part of QIAGEN in 2004, to work on new technology research and development, focusing on sample prep and diagnostic applications. In 2011, he joined the QIAGEN R&D team group in Hilden, Germany, to work on solutions for next-generation sequencing. Starting in 2015, he focused on microbiome extraction and associated workflows, and now leads the R&D group as Director for Microbiome Product Development. Dominic, are you with us?
Yes, Andrew. Hi. Good afternoon.
I can hear you great. Take it away.
Okay. Thank you for the introduction. And as Andrew mentioned, I'll be talking about QIAGEN solutions for microbiome analysis, specifically in the sample extraction portion of the workflow, as seen here. So I'm responsible for the development and optimization of the extraction kits used for microbiome applications. And specifically here today, I'll be focusing on the Power Soil and Power Fecal kits, both DNA and RNA focused. So as Andrew mentioned at the beginning, one of the things that we really focus on are stool, wastewater, sludge, also potentially swabs, soil, manure, all very difficult sample types. So these are some of the toughest sample types to extract RNA and DNA from. And in particular, when you're attempting to look at microbes in these sample types, you have multiple challenges. You have the challenge of lysing everything significantly, so getting all of the different bacteria with all the associated different difficulties of cell walls open, getting not only bacterial, but also viral and fungal nucleic acid out. But also these samples are very high in inhibitors. So all of these samples contain substances which are going to be problematic for your downstream extraction, your downstream analysis, if they're not dealt with during the extraction process. And for all of these kits, or all of these sample types, we have kits available. An example listing of them here is in the table. Starting off with DNA from stool or soil or biosolids, we always recommend the D&EZ Power Soil or QIAamp Power Fecal Pro kits. These are the same kits, use the same chemistry, are simply targeted one towards stool sample and one towards soil samples. And then we have the RNeZ Power Fecal Pro, and this does RNA or alternately total nucleic acid from also stool, swabs, sludge, and wastewater, and that's going to be the premier version to get RNA. We also have a variant of the previous two kits called the All Prep Power Fecal Pro, which will provide you with both DNA and RNA in separate eluates from those same sample types. In addition, if you're doing wastewater or potentially also open freshwater and dealing with very large volumes where you might have to filter the sample, the RNase Power Water Kit could be used in such a situation. It also still includes the IRT and will be able to deal with the concentrated inhibitors that you would get on a filter from doing things like filtering a liter or more of water. And the last kit that we have here on the list is the RNase Power Soil Total RNA Kit. And this is for RNA from soil samples, including from compost and manure, especially dealing with larger input amounts. And you can go up to multiple grams of material that you can extract RNA from. Now, I'm going to be focusing in the next couple of slides really on the Power Soil Pro and in the DNeasy and RNeasy sides. But the technologies that I'm talking about are implemented across this entire portfolio line. And so they are well suited for bacterial extraction from these very difficult sample types. So I was talking about inhibitors on the previous slide. And this is here just a schematic of what you're going to get. The issue with these samples from manure or stool or from the soil is that there's a very high level of inhibitors, things such as humic and fulvic acids, polysaccharides, polyphenolics, bilirubin, heme. All of these things have very strong associations with nucleic acid and also biochemically sometimes act very much like nucleic acid. Thus, they can be problematic for the sample extraction, where if you're going to be extracting your DNA, you may also co-isolate these inhibitors. And then for the downstream, if they act like DNA biochemically, then they're going to be problematic for your PCR, in that they may bind to your PCR enzymes and inhibit your amplification process. So it's essential that we remove the inhibitors during the extraction process before they have a chance to negatively affect the downstream application. And as an example, you can see here what happens if you take one of these samples. In this case, I believe this was a soil sample, and you prep it with and without the IRT that is present in our power kits. The eluate that you see on the left-hand side where it says inhibitors removed was processed with IRT. You can see how clear the eluate has come out to be. That same soil sample processed without IRT, but otherwise gone through the normal purification process. You can see how many impurities, just visually, you can see how dark the sample is and that the impurities co-isolated with the nucleic acid. And then you can see on the right-hand side in the PCR amplification curves, what a negative effect that has on your analysis. So when we use the IRT, and therefore the curves that are marked as minus inhibitors, so without inhibitors anymore, you can see very early detection around a CT of 12 or so, and perfectly normal amplification curves in your PCR. If the sample did not have the inhibitors removed, you can see in one case almost total inhibition with the amplification curve only coming up in the mid to high 30s. And in the other one, it's inhibited by a minimum of about 10 CTs. So it's a very dramatic effect on your downstream amplification if you are not able to remove these inhibitors during the process. So the IRT is an absolutely critical part of doing your sample extraction if you're dealing with these sort of difficult sample types. Another point, though, is that you're going to want to get the best possible yield out of the samples that you can get. And this is for particularly for bacterial, viral samples. When we're looking at wastewater, or if you're looking at swabs of hospital surfaces, if you're looking for environmental pathogens, in many cases, you're going to have a low starting material, and therefore the maximal yield that you can possibly get is very important. And here we have an example of how the Power Fecal or Power Soil Pro technology handles the yield from different samples. And this is one of the my favorite data sets because this is an experiment that was done on younger and older individuals and examining the effects of aging on the microbiome of the samples. And what we had is donor A and donor B were both older individuals, and the samples were much harder to deal with. They were more solid, they were difficult to lyse, and they performed very poorly in a lot of standard extraction kits, as you can see in the low yields from the two kits marked in blue, and performed very well with our PowerFecal Pro kit. Donor C was from a younger individual, and the sample was much easier to work with, provided generally good yields with almost any kit that we had. We got 2 to 3-fold better yields using our Pro kits on that sample, but the real major increase was on these difficult samples. On the right, you see the purity levels. You can see that the DNA coming off of these samples is highly pure for all of them. Now, the increased yield for these difficult samples is one important point, but what that really means in your downstream here is also interesting. So these are these same samples, samples A and B from the older individual, sample C from the younger individual, and this is a 16S NGS. So this is a V4 16S amplicon sequencing experiment. And here we're looking at the rarefaction curves and just looking at total number of species found. And you can see that we're using the Pro technology for the older individuals, really very significantly more total bacterial species were detected in the samples after the extraction with this new technology. In the younger individual, the extraction efficiency was very similar. So the easy to lyse sample, most kits were able to extract everything that was there. In these difficult samples, you really needed to have this Pro technology in order to see everything that was present in your sample. And when we drilled down into which bacteria were present in these, that was actually also very interesting because we saw in particular significant amounts of archaeal species as well as actinobacteria, so methanobacteriales and bifidobacteria, which are both known gram-positive stool community members, and those were dramatically better detected with our new kit, showing that that increased yield was really because we were detecting diversity that some of the other kits in the market are not able to find. So now I'm going to switch tracks a little bit and talk about RNA. So the RNeasy PowerFecal Pro is being launched in April of this year, and it is going to be a successor to the RNeasy Power Microbiome Kit. It's also designed for RNA extraction from stool and sludge and wastewater. And here you can see as well that the extraction of RNA that's our first bar, is the RNeasy PowerFecal Pro. Outperforms any other kit on the market, including our legacy kit. In this case, 4 different stool samples, 2 from adult and 2 from children. And again, giving significantly better overall yields, which can be very important, particularly for RNA sequencing, where the dynamic range is very high and having sufficient starting material can be very important. And then specifically for wastewater, we've also investigated the RNeasy PowerFecal Pro. In this case, we're looking for viral extraction, and we're looking for SARS-CoV-2 in wastewater. This is a surveillance activity that has become very important over the past couple of years. And here you can see the comparison of the RNeasy PowerFecal Pro with a workflow using the QIAcuity digital PCR system using the OAS advanced chemistry, compared to a workflow from Zymo using the Bio-Rad droplet digital PCR on wastewater samples from 2 different facilities. And you can see that the RNeasy PowerFecal Pro also had the highest possible detection of virus in the wastewater, showing that we get not just the best downstream inhibitor removal, but also the highest overall information content out of these samples. All right, and then my last 2 slides are on automation. So I've been talking about 2 example kits, the DNeasy PowerFecal Pro and the RNeasy PowerFecal Pro. And now these are also automated on various instruments. So we have automation on the QIAcube HT, but also on the QIA Symphony, and that's what's shown here. So this is the PowerFecal Pro, the DNeasy PowerFecal Pro chemistry, automated on the QIA Symphony. In this case, we also did an extraction, created 16S libraries, analyzed them with the Microbial Genomics Pro suite and CLC. And on the OTU table on the left, you can see the differences between 4 stool samples, 3 replicates each, manual or on the QIA Symphony, And you can see that we detect exactly the same bacterial species and the same composition, and that's borne out by the beta diversity that we see in the middle. So whether it's automated or not, you're getting the highest possible yield and diversity. And then as a last point, I'd also like to point out that all of the manual kits that I've been talking about are also automated on the QIAcube Connect. The QIAcube Connect in particular will automate everything after the initial beating of the sample. So from the IRT on can be automated on the QIAcube Connect to help you standardize and get better reproducible results. Right, so this is my final slide. Just to give an example, I've only talked about really two kits and with a bit of information that we have automation available for them, but the whole portfolio is quite broad. There's a broad portfolio of microbial sample disruption, multiple sample isolation kits in all the different formats that could be requested, and then going down to the PCR, digital PCR, and NGS workflow and into CLC. So for any microbiome workflow, there are solutions available from QIAGEN. And in particular here, of course, there is the AMR workflow that we're presenting today. And with that, Andrew, I can pass on to the next presenter.
Yeah, great. Thanks, Dominic. If you have any questions for Dominic, please put them in the Q&A section. I'm moderating the questions in the background, putting them into folders for each of our speakers. We already have about 7 questions for Dominic, so certainly this is an interesting topic for a lot of people. But let's just get a pulse check from people to see if this is an area that might be of interest to you and what specific areas you might be interested in doing some extraction technology on. So while you're answering that poll, let me bring up our next speaker here. So Dylan Barbera received his bachelor's degree in biochemistry and molecular biology with a minor in microbiology from Pennsylvania State University. He received his master's of science from Mount St. Mary's University in biotechnology and management. And Dylan began his career in NGS in 2015, joining QIAGEN in 2016 as a quality control scientist. He currently serves as our global product manager for QIAGEN's portfolio of microbiology and whole genome sequencing NGS products. So with that, Dylan, are you with us?
Yep, thank you for the introduction, Andrew.
Awesome, great. I will mute.
All right. So as you know, it was mentioned by Andrew, I'm here to speak to you all today about our newly launched QIAseq X-Hive AMR panel, which is our hybrid capture-based antimicrobial resistance NGS panel that just launched about a month and a half ago. So where we fit into the workflow is where any sort of QIAseq product fits into the QIAGEN sampled insight, which is between sample extraction and data analysis. What's included with our kits are So what's included with our kits, the X-Hive kit, when you order it, is a reverse transcription box that includes all of the components needed for reverse transcription, as well as the hybrid capture panel and all the hybrid capture-based reagents. The kit is used with our QIAseq FX DNA library prep kit, which is our standard whole genome metagenomics kit. So if you are starting with just DNA, which many people doing AMR are starting with, you can start directly at the QIAseq FX DNA library prep kit step. However, if you're interested in doing gene expression or in general working with either RNA or total nucleic acid, you can start with our multimodal single tube workflow in the RT step, which does everything necessary for reverse transcription of RNA, and then you move on in cDNA synthesis before you move on to general library prep and hybrid capture. Our kit features a 16-hour hybridization step, which is a longer hybridization step and is overnight. However, this is amenable to your standard kind of workday lab setup where you would do this beginning part in the first half of the workflow, and you would set up your hybrid capture overnight and then come in the morning and finish up and start sequencing the next day. So moving down the stream, you may ask why, to use hybrid capture in this sort of situation versus amplicon-based sequencing. So hybrid capture is amenable to much larger amounts of targets. Our panel specifically is for 2,786 unique AMR genes, but that's actually about 6,300 AMR alleles. So this panel is quite broad, and when you have a panel that is as large as this, typically hybrid capture is going to work a lot better for enriching for that many different targets. Hybrid capture is also a lot less susceptible to mutations and dropouts due to those mutations as compared to your standard primer-based amplicon sequencing. So if you have SNPs or deletions, hybrid capture probes aren't quite as affected, and you won't see quite as many dropouts as you would with an amplicon-based sequencing. This lower susceptibility to these issues allows for a much more comprehensive profiling of your targets and allows you to get much better data quality as compared to amplicon sequencing across a broad range of different species or targets that might have slightly different genomes. However, there are some cons to hybrid capture versus amplicon, which it does have a longer turnaround time typically, and typically requires more hands-on time. However, the benefit to me far outweighs the cost in that you do get a lot more information and a lot more data that's a lot less at risk of having any sort of issues to it. So as I think everybody probably calling in is aware, antimicrobial resistance is one of the largest talked about threats in medicine, where it's sometimes called the silent pandemic or the next pandemic, as there's many formerly treatable diseases that are becoming more and more infectious every day. So the US CDC has put out a list of what they determine as urgent threats, serious threats, concerning threats, and different things that are revolving around the world. So even in the United States, even in countries that have good access to antibiotics and other drugs, you're starting to see a growing number of deaths to bacteria and infections that are caused by antimicrobial resistance related bacteria. So in order to combat this, a lot of countries around the world and a lot of governments around the world are putting out funding for different AMR solutions or AMR research. Specifically, the United States CDC via the US Congress last year appropriated $172 million for AMR research. And this is actually something that you see around the world as well. Europe has a lot of funding for AMR research, as well as many countries in Asia and the general world. So at least for the US Congress-based funding, it is directly linked to all 50 state health departments and territory departments in the United States. And what they've established is the United States AR lab network, where it is a conglomerate of labs really working together to combat against this emerging threat. So our XHI panel is based off of our QMI AR database that is associated with our CLC Genomics Workbench. As I mentioned previously, it is for 2,786 unique antimicrobial resistance genes, and that translates to about 6,300 unique AMR alleles. What I have up on the screen right now is the subset of drug classes that the panel is protecting, or not protecting, but detecting against. And what that translates also is to a pretty broad range of different phenotypes. So our panel is pretty pan and antimicrobial resistance, where we're not looking at any sort of specific AMR mechanism. We're not just looking at perhaps beta lactamases or anything like that. It is pretty much every different phenotype of AMR that exists, where you're able to really detect any sort of AMR, pretty much any AMR. So you may ask, what is the benefit of this panel compared to current shotgun-based methods? So really the only main way to look at AMR at the moment using NGS is to do shotgun whole genome metagenomics, where you are trying to allocate enough reads on your sequencer and hope that you actually get your AMR sequences with the read allocation that you've given. Due to using this targeted enrichment, you're getting a much higher sensitivity, which is going to be able to detect those much lower abundance AMR genes, with a much higher level of enrichment, so this guarantees that you're going to capture your AMR gene of interest. What this actually also translates to is a lower sequencing cost when you actually move to sequence and try to capture your AMR genes and get data for them. An example for this is, if you are trying to do whole genome shotgun metagenomics on a MiSeq V2 or a MiSeq with V2 reagents, so you're doing whole genome there, and you are trying to make sure you get your AMR genes, you may only be able to run 1 or 2 samples on your MiSeq at a time using shotgun sequencing. However, with this panel, you are able to multiplex 14 samples at a time on a MiSeq with V2 reagents. So that is over 10 times as many samples can be multiplexed at once, which really saves you a lot of time in terms of doing your sequencing runs, and also saves you a lot of money on flow cells and other sequencing reagents. And because this is a targeted enrichment that you are comparing against a specific AMR database, we don't have to do that whole genome de novo alignment that can take a considerable amount of time if you're doing shotgun-based sequencing. So what this ultimately translates to is kind of depicted on the screen. So the top track that we have here is a shotgun-based sequencing of methicillin-resistant Staphylococcus aureus, and the bottom is hybrid capture enrichment. So what I want to highlight here is the highest level of coverage in the shotgun-based sequencing is 184, while with the targeted enrichment, it is over 9,000. So that is a pretty significant increase in enrichment of the AMR genes of interest. And that's largely due to the fact that you are not having to sequence areas of the genome that you don't really care about. So when you are just focusing on the genes of interest, and not focusing on this region here, you get a considerable extra amount of enrichment, and that really allows you to target those lower abundance AMR genes in your microbial populations. So also just extending to the different kind of sample types you can work with, what is depicted on the screen right now is 57 different saliva samples that were extracted with QIAGEN reagents and run with this panel and analyzed in CLC. So in the top, we have the different types of phenotypes that we were able to detect, which is a pretty broad range of phenotypes, and then a pretty diverse amount of different genes that we were able to detect in these saliva samples. On top of that, we also ran this on both wastewater and stool samples. Interestingly, wastewater, and kind of expectedly, wastewater is incredibly more diverse than just your standard stool samples in terms of antimicrobial resistance. Something that is kind of lost in the other here is we actually in the wastewater did detect genes for colistin resistance, where colistin is considered one of the last line of defense antibiotics that's out there and is used very sparingly. However, we are able to see the presence of this becoming more prevalent, or even prevalent at all, in wastewater samples in Europe. So that's kind of starting to show that perhaps even these last line of defense drugs, we're starting to be able to detect that even resistance to those is starting to come into the global population. And that is really important to try to prevent and keep track of and surveil to make sure that this kind of antimicrobial resistance doesn't continue to grow. So this is my last slide. And I just want to highlight again the various amount of different sample types that you can do with this type of panel. It's not really restricted to any sort of bacterial species. It's not restricted to any sort of sample type, and really allows you to get a broad amount of information and data from the amount or your sample of choice.
Great. Thanks, Dylan. That was wonderful. Let me just remind the audience, if you have questions for Dylan, please put them in the Q&A box. We've already got a number of questions teed up here for Dylan, so please continue with that. Let me just launch, again, just a super quick poll just to see if this is the kind of stuff that you're interested in learning more about that might influence us to do more of these kinds of programs and provide additional information. All right, so that brings us to our final portion of our presentation today, and that has to do with the analysis of the informatics. So our speaker is Daniel Hand. Daniel, can you maybe mute for just a second here while I introduce you? We're getting a little feedback. Thanks. So our speaker is Dr. Daniel Hand. Daniel is one of our excellent senior field application scientists at QIAGEN Digital Insights, which is the bioinformatics portion of QIAGEN. Daniel is based in the UK and did his PhD at the University of Birmingham. And during his postdoc, he became an expert in using DNA sequencing and phylogenetics to identify pathogens and derive drug resistance profiles in clinical specimens. So with that, Daniel, you can unmute yourself and take it away.
Thank you, Andrew. I'll just mute my slides. So you've already heard from the other speakers about our sample to insight workflow. And this talk, I will discuss the crucial final step, the bioinformatic analysis. How can we analyze these AMR hybrid capture samples? And this is an overview of what we'll talk about in this presentation. So I guess the end goal is to help you understand how quickly and easily you can analyze this kind of data. So I'm going to be going through a relatively small data set of stool and wastewater samples, including controls. And we're going to be generating some peptide marker abundance tables. So this is what we use to sort of calculate the AMR in a sample. And these are what we call shortbread abundance tables. And they're generated from a database that we use in the Genomics Workbench, which is the QIAGEN Microbial Insights Antimicrobial Resistance Marker Database. So when we have these abundance tables, and I guess you've already seen them on the previous presentations, we can start asking some questions. So we can start asking questions about what is our alpha diversity in our sample. So this is the diversity within a sample. So we can use that to answer questions like, have we done enough sequencing? We can compare between different sample types like stool and wastewater to see what the diversity is between the two types. And yes, we can start looking at the beta diversity to look at what is the difference between different sample types. Finally, we can put some stats to the analysis and start doing some differential abundance. And with this example, I'll move on to what's been discussed previously, looking for AMR markers for a key last resort antibiotic drug, colistin. So with that, just to show you where we're heading with this presentation. So on screen now, you can see the end result of an analysis in QIAGEN CLC Genomics Workbench Premium. So I've got 3 different visualizations and a QC report. So I'll just try and get my my annotation tools. So what you're seeing on the top left is these peptide marker abundance tables. But you can see that we have the samples arranged by sample type. So you can see on the left, we've got 3 wastewater samples. In the middle, we've got 6 stool samples. And on the end, we've got 2 non-template controls. And already with this phenotype view, we can see a difference between all 3 types of sample. We can look at the alpha diversity, as I mentioned, which is sort of the diversity within samples. And also, if we use the metadata, so in purple here, we've got stool samples, and in green, we've got the wastewater samples. We can see the wastewater samples are much more diverse in terms of AMR markers. We can look at the beta diversity to look at how the samples pull apart, and straight away we can see that the stool samples and the wastewater samples are pulling apart and are completely separate from the non-template control. And in the Genomics Workbench, you can see that we can also produce a combined QC report. And in this, what's shown on the screen here is the display for mapped human control genes, which we might expect in some samples such as stool. So moving on, just to give a bit of background, the product I'll be talking about today is QIAGEN CLC Genomics Workbench Premium. And within that, we have the QIAGEN CLC Microbial Genomics module, which is a comprehensive suite of microbial analysis tools, roughly broken into metagenomics, strain typing, and functional genomics. So in this talk, we'll mainly be looking at metagenomic analysis of AMR markers using those peptide marker shortbread analyses, but we could also do 16S and ITS analysis. If we had pure strains of bacteria, we could look for core genome and whole genome MLST. We could do other kinds of antimicrobial resistance analysis, such as SNP analysis or nucleotide gene analysis from de novo assemblies. Finally, we could do some functional genomics with annotation from imported databases. So in terms of the AMR databases we provide in the workbench, obviously you can import some of your own, but the main ones we provide are AresDB, which is an computationally inferred database, integrated database with peptide markers, SNP markers, and nucleotide markers, which you can see an example of. It's quite hard to see on the bottom of the screen there. But in terms of the relevance for this presentation, I'm going to be discussing the QIAGEN Microbial Insights Antimicrobial Resistance Database, where we have got a shortbread database of these peptide markers where we deduplicated and restructured public databases such as CARD and ResFind all in one place. And this is what the XHive panel, AMR panel, is also designed on. So you can see in the genomics workbench, we have a browser here where we can see what the database is made of and what the overlap is between the different databases. And not shown on screen there are some descriptions. So why is this database useful? If I show you the overlap here, can you imagine searching all 4 of these databases separately in an analysis? Well, you don't really have to do that if you use the QIAGEN provided database because we bring them together all in one place. So with that, I'm going to go into a live demo of how we produced this analysis in both GeneGlobe and QIAGEN CLC Genomics Workbench Premium. So I'll just share my screen with you. So when you conduct an NGS sequencing experiment, you generally end up with a folder of FASTQ files. So the question is, well, how can we process these in a simple and efficient manner to get some real results that impact our research? Well, we could, obviously, with the panel, you do get access to GeneGlobe. So you could use web-based analysis, and we have got a workflow on GeneGlobe where you can import those FASTQ files, and then you'll end up with analysis folders that you can download that will have those peptide marker abundance tables in an Excel format, but also in a CLC format. So I'll show those within CLC, Genomics Workbench. So in CLC Genomics Workbench, as well as generating those abundance tables, we can also go with the further analysis, such as looking at the alpha and beta diversity and the differential abundance. So just to show you, when you load up the workbench, you get a welcome page with example data and tutorial data to get you started. You have a comprehensive toolbox in the bottom left of the screen, which has template workflows and also the individual tools themselves. So if we open up the microgenomics module and then go into drug resistance analysis, you can find we've got our fine resistance with shortbread tool. So that's what you would use to analyze these data, these hybrid captured data, to look for those peptide markers. So running the tool is just as simple as double clicking it, and we have a simple wizard where we can go through the steps and generate those marker databases. So those peptide marker results. But I've brought together this tool and a number of other tools to do the alpha and beta diversity together in one workflow and installed it in the workbench here. So I've got an AMR workflow here, and we'll run that in just a second. So that is in read-only mode, so it cannot be edited by anyone accidentally. We could configure that if necessary at a later date. So to bring in, how do we do this analysis? How do we bring in the samples? Well, we go to import, and we've got a variety of NGS importers here. In this case, we would go to Illumina, we would go to add files, and you can see those are the FASTQ files we saw earlier. We would add them, and then save them to a folder, of which I've already done that in the workbench. At the same time, often with these experiments, we have metadata. So in this case, for the experimental metadata, we can create an Excel file and have our sample type here, because that's what we're most interested in this analysis. We just want to look at the differences between sample types. You can compare many different metadata factors or experimental factors in your analysis. So we would import this in by going to import metadata, and then selecting that Excel file. And then we can see it's in the workbench here. So to quickly start off an analysis, we would just click on the workflow here that's installed. We would select all of our samples and pass them just to the workflow. And then we would go through the options, of which I'm going to leave default in this instance. So in this intuitive wizard, you can see that we've already got the QMI AR peptide marker database, the shortbread database pre-selected. And then we've got some options for alpha diversity to look for diversity within a sample, and beta diversity to look for diversity between samples. And then we would simply select a folder to save the results in and click finish. That's as simple as that. Process into this tab, you've got an ongoing process with a log file, which we can start this process because we've already produced the results to save time for this presentation. So what do the results look like? Well, in terms of, I mentioned before, gene globe results, you get your peptide marker tables, both on an individual sample level, but also on a merged basis. So this is the kind of result that you would get if you imported it into a free version of the workbench or the premium version of the workbench. But in terms of going further with that, what does the workflow produce? The workflow produces both these individual reports in folders for the shortbread tables, but it also produces a merged abundance table here. And it also produces the ongoing results in terms of alpha diversity, rarefaction curves, beta diversity, heat maps, and combined QC report. So if we were to look at the stacked abundance results, you can see that we have some options here in terms of we could look for the feature we want to aggregate against. So in this case, we can change it to phenotype. So that's very similar to what you saw in the illustration earlier, or we can change it to gene name. Because we've got the metadata already in, we can paint this onto the samples or organize this onto the samples. So we can sort samples by sample type, which was what was in that Excel file. And now we can see we have 3 wastewater samples here, 6 stool samples here, and the non-template controls, and they already look very different. So we're already getting some of those insights. And as Dylan mentioned, you can see the incredible diversity that's present in the wastewater samples as compared to the stool. Well, at the sort of the higher fractions, they do look a little bit similar, which might be what you would expect. We can open up our alpha diversity, and again, we can do the same thing. We can look by sample type to look at the stool samples are indeed a lot less diverse than the wastewater samples, and we can open up our beta diversity. And then in a similar way, we can look at the metadata and change this to sample type, change the text to sample type, and then you can see intuitively how the samples are pulling apart based on principal component. Going further than this, so we've already produced some results and seen that there's a difference between the samples. How do we go further than this? Well, we can put some stats to it and run our differential abundance tool. If we were to do that and search for differential abundance analysis, we can select this, and this will allow us to select an abundance table, so our final peptide AMR abundance table. And then because we've got the metadata associated, we can use sample type to look for the difference by sample type, and then perform an all groups pairs comparison, and then have a look at the results. And we're interested in stool and wastewater. And this one, we can organize it by FDR p-value to look for the most significant differences between stool and wastewater. And if we look at the heat map as well, and just also add the metadata to this, so it's just a little bit so you can see our hierarchical clustering is already pulling apart samples. We can look for the most differentially abundant AMR marker profiles and then select this on our table here. And you can see we can highlight these markers on the heat map as well, and it does correspond with the intensities as we'd expect between stool and wastewater. Okay, so hopefully I've shown you how quickly and easy it is to produce a range of different analyses. So to finish off, I will move back to the presentation and move on to the colistin example Dylan was talking about. So how can we ask a really interesting question of this data? And if we look at that differential abundance, can we identify a marker for colistin resistance, MCR1? How does that look in the software? So in this, if I just show you the end result of this kind of analysis, you can see at the bottom, we are looking at that QIAGEN QMIAR database, interrogating it for MCR1. And we can see that we've got a plasmid-borne and a chromosomal-borne MCR1 markers in the database. We can put this into a filter on our differential abundance table. And actually what we can see here is we've got we've got a statistically significant result showing that last line result antibiotic marker is present in the wastewater, but not the stool. So we're using the wastewater as a sort of a surrogate for the larger population. So hopefully you found that interesting, and I'll show you how quick it is to analyze this kind of data in both a web-based application, GeneGlobe, and a desktop application in QIAGEN CLC Genomics Workbench. And you can see how you can apply it to your own analyses. With our products, we offer full tech support and also tutorials to get you started. And with that, I'll move to questions and hand over back to Andrew.
Yeah, great. Thanks so much, Daniel. Please, again, put your questions for Daniel and our other two speakers, Dominic and Dylan, in the Q&A box. And while you're doing that, let me just launch the last of our quick little polls just to see if this is a topic that you'd like to see more of and would like to learn more about. We have a number of questions here for each of our speakers, and I've received a number of questions about, will this recording be available? Absolutely, 100% yes. We'll be sending out, or you should get an automated message with a link to it here in the next couple business days that you can go back and relive this extraordinary event. All right, with that, let's go ahead and dive into our Q&A. And let's see here. I'm going to start here with Dominic. And first question for you, Dominic, is for the best extraction yields and purity while using the Power Fecal or Power Soil Pro kits, is it best to use the samples while still containing the water content, or can the samples be air dried prior to lysis and extraction? Does the water content present in the sample impact the success of the kit?
I would say yes in some extreme cases. So generally we work with natural samples, so we do not dehydrate the samples prior to processing them, because as soon as you dehydrate them, you're also going to be potentially changing the bacterial composition that's there. Some samples will be happier with lower water content and some will not. We tend to process them as they are. The first step is always to add a lysis buffer. So we do our bead beading in a liquid. And so you have to have sufficient liquid for the bead beading to occur. If you dry your sample too much, it could absorb significant amounts of the liquid. In that case, all you need to do is add a bit more of that first lysis liquid to get up to the same overall volume, and then it will be no effect.
Gotcha. Great, thank you. Our next question here is for Dylan. Are AMR genes covered animal pathogens also apart from human? I think what she means is, does it cover animal pathogens in addition to human or aside from human?
Yes, it's not specific to pathogens that are just from human or human infections. So as I mentioned, it is based off of that QMI AR database, which Dan went over, the big conglomeration of databases that is putting together. And those are not limited to human-only AMR bacterial genes.
Gotcha. All right. Next question is for Daniel. What kind of data input is allowed into the CLC microbial genomics module?
So in terms of sequencing types, we support the major sequencing technologies. So in this case, it was Illumina. So it was FastQ, but we also have importers for Oxford Nanopore, Ion Torrent, MGI, to get the next generation sequencer data in, if that's I guess that would be the main data type that you'd want in. But we also have, you can import other types such as track data or region data that are associated with chromosomes and that sort of thing, and a whole load of other data types. But the main one would be those NGS data importers, and we cover the major types.
Gotcha. Let's see here. For Dominic, the question is the thinking that these kits can be used with simpler specimen types?
Yes, absolutely. Yeah. So the kits are I guess, downwards compatible, if you will, right? You can use it for swabs or low biomass samples, and it will work very well, and will still do really good bacterial extraction. But you can't go the other way. You can't take a sample that does not have an inhibitor removal technology and use it for something like sludge or wastewater, since it doesn't have that key component. So,
got it. Okay, great. Question here for Dylan. Is a comprehensive list of the targets available? Possible wondering about the coverage for the various CPE genes.
So the list of the targets of the panel are on the QIAGEN.com product page, and this also includes a list of all the viral targets we have with our X-Hive viral panels. It also can be provided by your QIAGEN rep, but we do provide that full list of genes that are targeted.
Gotcha. So next question, I guess, is for Daniel. It's kind of a generic question, but apart from CLC genomics, can we use other bioinformatics pipelines to analyze the data? I guess my answer on that would be, we're not experts on other tools, but I don't know. Do you have any input on that, Daniel?
Yeah, of course. I mean, there's a variety of, I guess, open source tools and other vendors that you could analyze the data with. Hopefully, I've highlighted in today's presentation what are the benefits from using our solutions, but of course, you could analyze the data.
Sure, and I think maybe we glossed over it a little bit. It's my understanding that our hybrid capture kit comes with the ability to analyze your data in the GeneGlobe application without additional cost. Is that correct?
Yes.
Maybe that's a question for Bill. Yeah, sorry.
Yeah, so that I guess we didn't fully expand on. So our X-Hive panels can be analyzed, included with the cost of the kit on our GeneGlobe analysis portal. And what that provides you with is a list of the different AMR genes detected and what drugs they confer resistance to, as well as how many reads are associated with that. And then for a more in-depth analysis, you can use the CLC Genomics Workbench. However, there is free analysis available with the GeneGlobe analysis tool.
Great. We only have time for maybe another 3 or so questions. So a little off topic, but question for Dominic. Which purification or reagent methods would you recommend for samples from skin swabs for the study of skin microbiome samples?
I would also recommend the Power Fecal Pro, so either the Power Soil Pro or the Power Fecal Pro. I always recommend both because they're the same kit. I mentioned that during the talk, but bears repeating. So either one of them work very well with swabs. We would tend to recommend working with the nylon rather than cotton swabs, and not to use transport media if possible, since not all transport media are compatible with the chemistry.
Got it. And let's see here. I'm just trying to pick out a couple other ones here. I think this is more for Dominic. What is the sensitivity of hybrid capture AMR panel? How low abundance AMR can be detected and sequenced?
I think that's probably more for me. So, I mean, the materials, I mean, I guess it's kind of hard to specifically quantify. So the panels, in terms of when you're working with like the viral panels, are good down to 10 copies of actual viruses. And then this is compatible with for AMR, with our FX DNA library prep kit, which is good down to 20 picograms of DNA. But really what it results in, what you need is an actual like output from your the PCR in your library prep. So, you know, that can be kind of amended, that you do a few extra PCR cycles to amplify a little bit more. So you can start with as low as, you know, 1 nanogram of DNA, but you do have to, or even lower, but you do have to do more PCR cycles if you're not quite getting the amount of library you need to enter into hybrid capture. And the amount of, like, the concentration that you need is outlined in the handbook, and, you know, this extra amplification, I believe, is outlined in the handbook as well. But for bacteria, it's a little bit harder to quantify the amount, since you can just kind of amplify a little bit longer.
Got it. Okay, and then I think we'll see, this will be our last question here for Daniel, and I apologize to the participants if we didn't get to your question, but we have just so many here, we just can't get to every single one. The question is, do you have any pointers to do more in-depth analyses? Oops, I just lost the question. Here it is. More in-depth analyses by including metatranscriptome data and correlating expression patterns of the AMR genes.
Okay. Yes, I mean, we've recently released with the latest version of the Workbench some metatranscriptomic analysis tool, so that can help you out with that. Obviously, you can do your AMR analysis as well. In terms of correlating them, I guess on the fly, I guess I'd have to think about that. You can extract your, what I didn't show you because it would be too in depth. You can go into some of these workflows and modify them so they produce the sequences at various stages, and then perhaps you could do some sort of comparison of those two. I mean, I had to think about exactly how that would work, but yes, on its basic level, we do now offer a metatranscriptic analysis tool.
Maybe that could be a topic for a future webinar solely on that topic.
Yeah.
So great. All right. Well, I've put up a slide here of contact information should you have any questions. I apologize. The Digital Insights has a capital I for insights. It's not double L. I apologize for that. My apologies there. Anyway, so that concludes our talk today. Again, I apologize if we didn't get to your question, but hopefully this was informative, and we look forward to talking to each and every one of you in the future. And that concludes today's webinar. Thank you so much.